In our last videos, we gave a tour of the brain microtubule, and we even got to run our first shockwave experiment and vacuum. Today we will be running a series of realistic shockwave experiments by introducing water. This simulation type is called an explicit solvation, meaning that we introduce our structure to water. Here we have about 270,000 water molecules, visually represented as points, since it makes rendering this video faster. We are also using a smaller protein assembly since it is very computationally expensive to simulate millions of atoms. The purpose of these simulations is to see if it's possible to replicate CTE conditions, where the purple-colored tau can separate from the green-colored tubulin mechanically. When this purple protein accumulates, depending on how it's folded, it can lead to brain disorders like Alzheimer's disease, chronic traumatic encephalopathy, and various other taupathies, such as frontotemporal dementia or Pick's disease. Now let's begin with a simple single shockwave. This isn't a regular shockwave, but a hypersonic shockwave created by a virtual piston, traveling at about 12,000 meters per second, slamming into our brain protein. Even with the strong shockwave, our structure seems to be intact, despite some bottom edges shaking loose and the middle section slightly separating. Let's add some randomness to the shockwaves now. Under a barrage of multi-directional shockwaves, the microtubule undergoes heavy damage. The rigid green tubulin are driven outward while the tau proteins are stretched to their limit, barely clinging on. In the time it takes you to blink once, one of these shockwaves could travel the length of 10 football fields. Despite this incredible shock, the tau is resilient due to the last contact points having unique anchoring properties. You are watching a dual-piston ultrasonic press. We've programmed the water at the top and bottom of this box to vibrate at 60 billion times per second, effectively hammering the protein from both sides simultaneously. Because water is highly resistant to compression, this force is transmitted directly into the protein, creating localized pressures that can exceed 20,000 atmospheres in just a matter of trillionths of a second. Despite extreme compression, once again, the purple tau proteins remain attached thanks to their microtubule binding region. While these green-colored tubulin proteins have highly negatively charged surfaces, the binding region of these purple-colored proteins are packed with positively charged amino acids, creating a strong electrostatic bond. Think of opposites attract. You're probably wondering what happens next. Our bodies actually have a dedicated cleanup crew designed to identify damaged proteins and grind them down into reusable amino acids. This is called the ubiquitin proteasome system, and specialized cells called microglia. The issue here is that this cleanup system can easily get clogged. Imagine throwing a bundle of sticks and glue into a paper shredder. The sticky tau and the fragmented tubulin create a jam that the cell can't easily clear. So why is this important? Well, in neurodegenerative diseases known as taupathies, tau stops binding to microtubules and begins sticking to itself instead. While we don't see the full accumulation in the specific simulation, the brain is vulnerable to a process called pathological seeding. This is a domino effect where a single misfolded tau fragment forces healthy tau proteins to misfold and aggregate, spreading the disease across different brain regions like a wildfire. Ultimately, while the long-term formation of CTE is driven by chemical changes like hyperphosphorylation, the process often begins here, with the literal physical stretching and shattering of the neuron's infrastructure. Join us in the next simulation where we're getting our hands on the actual pathological brain protein harvested from the diseased brains. We're going to even deploy a nanorobot into the accumulated tau and see if we can mechanically save the neuron. Thanks for watching.